All right. Hi, everyone. My name is Jason Gomes, and as Matt said, I'm from the Community College of Allegheny County. And today I'm going to be talking about T-cell and CAR-T therapies in human cancer treatments, as well as their potential translative application in animal health. Don't worry if you have no idea what any of that means. Hopefully by the end of this presentation, you'll have some understanding. Before we get into the what and how, I'd like to discuss the why. Why did I do this research, and why does it really matter in the context of community? I was first introduced to T-cell receptor technology when I interviewed for a company that was actively working to develop new TCRTs in order to combat specific ocular cancers. Since then, I began to notice more and more pharmaceutical companies focusing on this technology and the closely related chimeric antigen T-cell receptors, or CAR-Ts. So clearly there was something to this. That's what I wanted to find out. As an honor student, I was given a chance to do a research project, and as someone seeking a degree in the biological sciences, I thought, what a better time to research. So I wanted to answer a few questions. What could they do for cancer treatments in humans, and how would they be effective in those cancers? Further from those, as an animal lover who has 4 cats, a dog, and a few rats, I'll show you guys some pictures at the end if you're still here. I wanted to know if these treatments could translate to treatments in our pet companions. As you'll learn in this presentation, there is some great potential, at least in canines, to apply these new treatments. And I was surprised, in fact, to learn some of what I'll be presenting. Now let's talk relevance. Why is this important to community? According to the National Cancer Institute, in 2018, there was 18.1 million new cases and 9.5 million cancer-related deaths worldwide. I think the relevance there is fairly simple. That's a large chunk of our community affected. As one of the leading causes of death around the world, it's understandable that there are many researchers looking into new therapies to find a cure. Based on the research I conducted, TCRs and CAR-T therapies are showing promising results in both preclinical and clinical studies. In one article I read titled "CAR-T Cells Driving the Road from the Laboratory to the Clinic," the authors go so far as to state, "The impressive objective clinical responses reported in results of trials testing antibodies, cytokines, and immune cells against advanced cancer are fueling an intense review of immunotherapy and the development of a range of immune-based tools that are becoming increasingly available to clinicians." The impressive clinical responses certainly explains why I was seeing an upward trend in trials testing these therapies. So what exactly are T-cells, TCRTs, and CAR-Ts? Well, we won't be getting into the nitty-gritty of the immune system and specifics of how T-cells interact with other cells. A basic understanding is necessary for this presentation. T-cells, also known as T lymphocytes, are key components in the human immune system. There are 2 types of T-cells, helper and cytotoxic. The role of helper T-cells is to call other immune cells to attack an antigen being the foreign substance that the body is responding to. These other immune cells that it calls could be B cells or even just cytotoxic T cells. The role of cytotoxic T cells is to create a pore in the antigen presenting cell, or APC, then deliver what's called granzymes in order to cause the APC to undergo cell death, referred to as apoptosis. Cytotoxic cells are responsible for the cellular response to virus-infected and cancer cells, so that's largely what we'll be focusing on. So how do T cells work? When they leave the thymus, T cells have unique receptors which will only fit on specific antigens. So for instance, a T cell which can attack the flu will have a different receptor than a T cell that attacks the specific lymphoma. Another more recent example of this is when scientists talk about changes in the COVID proteins and why things like the vaccine are less effective because the protein has changed. Essentially, new T cells are needed. T cells will recognize the antigen based on a 2-chain protein receptor called an alpha-beta chain, which is pictured here. The point of TCRT is to transfer an alpha-beta chain onto a T cell in order to endow it with antigen specificity. This process helps to overcome the lack of natural antigen-specific T cells in the human body. There are a couple of downsides to T cell technology, however. The first is that they have a restrictive nature and can only be of use in patients who have the specific human leukocyte antigen. This in itself is the restrictive nature of naturally occurring T cells as well. The second is that tumors present a downregulation of antigen molecules as part of an immune escape strategy. In essence, when the immune system is responding to the tumor lessens the number of antigens it's presenting. As a response to those downfalls, researchers developed chimeric antigen receptor T cells, or CAR-Ts. CAR-Ts are synthetic receptors that have a modular nature, which allows them to usually redirect T cells to tumors independent of the human leukocyte antigen being presented. This ability allows them to overcome the restrictive nature that plagues standard TCR technology. One researcher even refers to them as living drugs which could obtain what they called supra-physiological possessions and performance. As impressive as these sound, there are, of course, some downfalls and risks in CAR-Ts as well. According to an article titled CAR T Cells in Solid Tumors, Challenges and Opportunities, which was published in Stem Cell Research and Therapy last year, when targeting solid tumors, it is more common to target tumor-associated antigens, wherever the expression of specific markers are increased. However, the markers that are targeted also occur at low levels in the body's natural tissue. This natural expression of these markers leads to something known as on-target off-tumor toxicity, which means that the CAR T cell attacked the right mark, but not on the tumor. This could cause extensive damage to the natural tissue, and obviously that wouldn't be the desired outcome. Additionally, they note that this method of targeting solid tumors make CAR Ts less effective in treating those as opposed to blood cancers, which have different markers. Now that we kind of have a basic understanding of how these things work and what they are, I'm going to dive into the research that I found in human health before we move on to how it can be applied in animal health. For the research paper that I wrote itself, I was required to find and cite at minimum 6 peer-reviewed primary sources. And primary just means the sources needed to have conducted the research themselves. While I used several other non-primary sources in order to write the paper, I'll be focusing solely on the primary ones. To find the sources used, I primarily utilized the database called EBSCOhost, which I receive access to via CCAC. I found and used 1 phase 1 clinical trial and 3 preclinical studies. Finding published primary sources on this specific topic ended up being fairly difficult, and I assume that's kind of due to the relatively new nature of the technology. In the phase 1 clinical trial conducted in 2018, researchers conducted a study on 4 patients, I know it's small, who had the New York esophageal squamous cell carcinoma antigen, also known as NY-ESO1. In the trial, 2 patients had a clinical response to the treatment. While it was an extremely small trial, a fact which was noted by the researchers did conclude that based on these results, TCR-T treatment showed promise in treating NY-ESO1, and that more large-scale studies should be conducted. This was also the only primary source that focused on TCR-T and not CAR-T. In the first preclinical study used, which was conducted in 2016, researchers were again focusing on NY-ESO1. However, this time they were looking to utilize CARs to target a specific antigen and peptide, which was only expressed in these tumors and that was not found in normal somatic cells. In the initial testing, they found that the CARs would lyse non-specified antigens and therefore had to change their method of producing the CAR in order to improve its specificity. They found that 1 mutation caused the specificity and concluded that these results showed exciting potential for CARs to expand their repertoire to include a broader range of antigens. In a second preclinical study, which was conducted in 2007 on murine models or mouse models, researchers sought to use CARs in order to enhance the T cell anti-tumor activity and address the limitations of failed T cell co-stimulation. They found that the use of CARs containing the proper signaling domain did indeed enhance the anti-tumor activity and noted that modifying the CARs in conjunction with T cell dosing allowed for complete eradication of systemic disease. The last source I found for human research is slightly different from the others. Earlier in the presentation, I mentioned that CAR T cells had some issues, especially with solid tumors. And this study, conducted in 2019, sought to apply chimeric antigen receptors to a different type of immune cell known as natural killers, which would complement CAR T therapies. They hypothesized that given their different homing properties, CAR-NK may be complementary to CAR T and potentially safer, which would result in less on-target off-tumor toxicity. In the study, they developed a new virus-free way to create these CAR-NK cells and found the therapy to be highly efficient and avoided risk associated with viral vectors. So the research all shows promising results in humans. But let's be real here, if you're an animal lover, you came here to hear about how it can help pet companions. While conducting my research, I was very surprised to find that there was very little published around utilizing these technologies in animals. In fact, finding the 3 primary sources I did felt like I found the only 3 primary sources. Though it should be noted that I was able to find some research around application of TCR in CARs in treatment of other diseases, just not cancers, which was my focus. The sources I found focused on application in canines, and finding research in felines or other animals outside of mice proved extremely difficult, almost impossible. So I'm going to be focusing on how it can be applied to canines. While primary sources were hard to come by, I did find several secondary sources that discuss the similarities between cancers in canines and humans. According to these sources, cancers in dogs share many features with human cancers. One surprising similarity is that they share many of the markers and the same antigens that we see in humans. So theoretically, treatments in one species could directly translate to the other. Additionally, there are an estimated 1 million dogs diagnosed with cancer in the US each year. Due to the abundance of canines diagnosed with cancer and the similarities between them and humans, there's a long history of dogs in biomedical research. Further, it's noted that the interaction between T-cell receptors and antigens in both share the same restrictions. So with that in mind, it was rather surprising how little research I could find. The same method for finding these research articles was used as locating the human research sources. I was able to find 3 preclinical studies. In the first study, conducted in 2016, researchers sought to create a new animal model that would better reflect human biology. They noted that this was needed in order to progress the research in immunotherapy techniques. Through this study, researchers found that the dog leukocyte antigen, the canine version of the HLA we discussed earlier, shared striking similarities with the human antigen expressed in NY-ESO1, known as HLA-0201, which is the same antigen we discussed. The findings of this research opened the door to applying T-cell therapies to dogs because prior to this, only a small amount of data had been available. The second study sought to develop a system that would enable us to evaluate CAR T-cells in dogs with spontaneous cancers. They found that the CAR cell in dogs with B-cell lymphoma was well tolerated and showed a modest anti-tumor activity. This study laid the foundation for use of outbred canine cancer patients in research of immunotherapies. The third and final study was one that looked to optimize the manufacturing of CAR T cells against canine B-cell lymphoma. Conducted in 2020, this research established a virus-based canine CAR T cell generation method and provided the fundamental data for better understanding of canine adaptive T-cell therapy. As you might be able to tell based on these sources, research for these treatments in canines is much further behind than human research, with much of the current research laying the foundation still. This fact is rather surprising when you consider that treatments often start in animal models and then move to humans. Perhaps this is a testament to the potential of TCR and CARs to treat and cure cancers. So, what research is currently being conducted on these treatments? According to clinicaltrials.gov, as of October of 2021, there were 435 actively recruiting human clinical trials that contain the keywords CAR T or chimeric antigen receptor. So, given the similarities between human cancers and canine cancers, one might expect that there would be an equally large amount of trials being conducted in animal health, right? Unfortunately, I did not find that to be the case. While there is no central database for veterinary clinical trials, I searched across several researchers and colleges and universities and was only able to find 3 actively recruiting trials. 2 were being conducted at the University of Pennsylvania and 1 at the University of Wisconsin. In conclusion, TCR and CAR-T therapies show a great deal of promise as treatments for many different types of cancers. And I believe that that perceived value is shown in the great number of ongoing trials being conducted in human health. However, I find it disappointing that there's a lack of volume for these studies in the animal health arena. Advocacy for translational research should continue as more breakthrough therapies are discovered, and the number of translational studies should be ever increasing. The increase of research can only be seen as a net benefit for both animal and human medicines. And as promised, here are the pet pictures. Any questions?
Can we see those pet pictures again?
What's that?
Can we see those pet pictures one more time?
Oh, absolutely.
Those are great. I think your line of, sort of line of logic and, the way that you present this is really, really helpful. For someone who's not in this field, it's really digestible. It's definitely a solid argument that you make here.
Thank you.
What do you think are some of the reasons or barriers to, that such a discrepancy between the humans and animal treatment, or even the research into it?
Yeah, so I think a lot of the barriers stem from one is funding. Animal health obviously doesn't get a whole lot the same amount of funding that human health does. And second, I think there is obviously a push towards taking animals out of the human health pipeline, so to speak, and using them as guinea pigs. But in this specific case, I think it's a therapy that's beneficial to both and should be applied to both and tested to really advance it.
All right, and I guess if there were to be an increase in testing in canines and other animals, I'm assuming that the benefits ultimately would benefit kind of equally the animals and the humans. It's really a symbiotic relationship with research. Is that correct?
Agreed, absolutely. Like I said, theoretically, given the similarities of the cancers between humans and canines, advancement in one should be able to directly translate to advancement in the other.
Any other questions from our audience? We have a few other participants in the Zoom session right now.
I came a little bit late because I was having some trouble logging in, but I don't know if you went over this in the beginning, but do you have like a personal connection to this topic?
So I have professional experience in the clinical trial and like biotech industry, and just noticed that CAR-Ts and T cells were on the rise for being tested. And then, as you can see from the screen, I have quite a few animals. So I wanted to understand how that therapy that was being tested so rigorously might apply to actually help animals as well. Any other questions?
Jason, thank you very much for going first and presenting on the topic. Super interesting. I have nothing to do with the field, but it really makes me want to learn more about it. And there's definitely so many potential benefits from this. I hope you're able to take it further.
Wonderful. Thank you. Thank you very much.
So if you could, stop sharing screen, and I'm gonna. Elizabeth, it's good to see you on the call. I'm glad you worked out whatever tech issues you're having before. I'm going to make you a co-host. Okay. And so our next presenter is Elizabeth Kagan from Macaulay Honors College at CUNY, and her topic is disrupting the stigma of medical marijuana use and treating maladaptive behaviors in individuals with ASD. Elizabeth, are you ready?
Yes. Let me share my screen. Okay. Sorry, I'm trying to find the right tab. Okay. Okay. Can everybody see my screen? Okay. All right. All right. So hi, everyone. My name is Elizabeth Kagan, and for this year's conference, I will be talking about a field of autism research that has been getting some traction in recent years. And this field is the use of medical marijuana as a treatment for the maladaptive behaviors in individuals with autism spectrum disorder. Okay. My connection to this project. So before I begin my presentation, I wanted to briefly explain my connection to the project. I have a younger brother who has autism. He is a year younger than me and recently turned 21. I have always been his overprotective older sister, as you can see in the picture of us when we were kids. And he has been my inspiration for pursuing this topic. And I just wanted to share the knowledge that I learned through my research with all of you. What is autism spectrum disorder? Well, the textbook definition is that it is a developmental disorder that impairs one's ability to communicate and interact, at least according to Google. However, it is hard to narrow down autism into one succinct definition because it is a spectrum disorder. What this means is that each individual will vary in the severity and onset of its symptoms. Some will have mild symptoms, while others will exhibit symptoms that will impact their ability to function more severely. There is no cure for autism, as it is a condition that one will carry throughout life. However, that does not mean that just because there is no cure, this does not mean that there are no treatments available for its symptoms. Okay, so currently there are numerous treatments, services, therapies, interventions, and other resources which are available for individuals with autism. Some of these help improve speech and language skills, while others help modify behavior, and still others improve cognition and learning abilities. The end goal of all services is to help these children grow to become less adults, which are less reliant on others in order to function on their own in society, at least to the best of their ability. Many of these are familiar to me as I watched my brother go through various special education programs, speech therapists, psychiatrists, etc. However, my brother is higher functioning, so his main obstacle has always been his communication deficit. Other individuals may require different treatments according to their personal needs and their symptoms. So I just wanted to share this quote by Dr. Stephen Shore because I believe that it greatly emphasizes my previous point. He says, if you've met one individual with autism, you've met one individual with autism. Just as neurotypical individuals are all different from one another, such is the same for individuals with autism and any disorder. People are individuals and they need to be treated based on their individual needs. Because of the barrier with communication, it is pretty common for individuals with autism to exhibit maladaptive behaviors. These can range from non-compliance to aggressive behaviors to self-injurious behaviors. all of which can be very challenging and problematic for the child and parents. While the current behavioral treatments which are available might work for some individuals, there are still others who require different approaches and treatments, some of which may not be so easily accessible. So now we'll get into the medical marijuana part of it. In recent years, families of children with autism have piqued an interest in using medical marijuana to alleviate various autism-associated symptoms. In many states, medical marijuana is being researched for its use in treating various health-related diseases and conditions, some of which happen to be comorbid symptoms of autism. This crossover is really interesting because it shows that there is potential for this treatment. This map highlights the legality of medical marijuana across the United States as of February 2022. In the states that are shaded in the dark green, medical marijuana is legal. Medical and recreational marijuana is legal. In the light green states, only medical marijuana is legal. And in the unshaded states, marijuana remains illegal. So moving on to the current state of autism and medical marijuana research. As you can see, it's still in its early stages. So most of the information out there comes from 5 small studies and numerous accounts of individual family experimentation. And one of the biggest hurdles in this treatment is that marijuana is still classified as a Schedule I drug, according to the FDA. Therefore, there is unequal access to it across state borders, and using it would be considered a felony in some states. All right, so while I'm now going to get into all the case studies and personal experiences of families trying medical marijuana, I just wanted to bring up one case of a young girl named Kara from Texas. Kara has a case of autism with very severe, dangerous, and violent fits. These include self-injurious and aggressive behaviors, such as throwing objects, breaking things, hitting others, and closed-fisted punches to the face. One night, she repeatedly hit herself for 12 hours straight. These behaviors are repetitive, which is a part of the disorder. She started to exhibit these behaviors around the age of 4. Her parents even got her doctor to prescribe psychotropics, which are typically used for schizophrenia, but these drugs left her unable to do anything. She was not present or aware of her surroundings, and she was on the highest dose, but she was still hitting herself. She would hit herself 3,000 times in a 7-hour school day. In 2009, Kara's parents were inspired by the story of a boy named Jay who had ASD and tried medical marijuana to treat his maladaptive symptoms. However, because Texas has very restrictive cannabis laws and it's illegal there, her parents still wanted to try it. So they decided to try marijuana brownies. When Kara first tried it, she had stopped hitting herself, was sitting still, looking out of the window and smiling. Kara's parents were in complete shock and disbelief as they had tried everything and no other treatment or medication had been this effective. They continued to go behind the law to give her marijuana. However, in Texas, the search for cannabis that is potent enough to stop Kara from punching herself is quite challenging. Her parents have to go to an underground source to get the weed, and they can't really work with many strains or doses to see what works best. They use a 1/4 teaspoon dose through her marijuana machine, which is a vaporizer which heats up the cannabis so that she can inhale it. Kara is eager to inhale the cannabis, and its effects are immediate as she starts to settle down and clap, seeming to thank her father for giving it to her. Her mind and body slowly relax and calm when she inhales marijuana. While it does not stop all of her autism symptoms, it definitely helps as she is happy and feels much better. Even though there are barriers that still need to be overcome within this field of research, the few studies that have tested it have found promising results. Individuals with autism had less behavioral outbreaks, communication problems, anxiety, stress, and disruptive behaviors. There have also been cases in which individuals reported social improvements and overall quality of life improvements. In summation, more research still needs to be done in order to have more evidence of the benefits that it can offer to individuals with ASD. And I'm not saying that this treatment will work for everyone, but it can really benefit those who need it and can't access it or don't even know that it exists. Okay, thank you. This is the end of my presentation. Does anybody have any questions? Okay. I don't know, just leave it.
So I have a question.
Mm-hmm.
So have you found that medical marijuana helps your brother?
So my brother never tried it. His symptoms are not so severe where my family has felt that we needed to try it. However, there are still individuals who have more severe symptoms where they may not, no other medication may be as effective as this for them. And because there's still like the issue with legality across the United States, it's harder for some families to have access to it, as well as because it's in its early stages, like it's not very well known. So
Right.
I think it's important to bring awareness to this field of research.
Awesome. Thank you.
Yeah, no problem. Okay. Anybody else?
Elizabeth, our computer died during the middle of your presentation, so I'm sorry I missed the end of it, but it was super interesting.
Thank you.
Thank you for, I don't know if I missed other questions, but thank you for letting us into your family and your what inspired you to really pursue this field.
Yeah.
Can you summarize the conclusion or the very end of it for me?
Yeah, it basically the research is still in its early stages and it has some promising results. There still needs to be more research done on it since this can be a potential treatment for certain individuals that may require it. And some people might just not have access to it, but because they're living in a state where marijuana is illegal, or they just don't know that it exists.
Gotcha.
Yeah.
Any final questions for either Elizabeth or Jason today? All right, well, thank you both for presenting this virtual format today. I hope that you get to interact with other parts of our conference the rest of today and tomorrow. And yeah, thanks again.
Thank you, everyone.
Thank you.
Have a great day.
You too.
Good.